Hi, I'm Marcela Ishihara, and I'm a PhD student at Butantan Institute. And today at this post, I'm going to talk about a new platform developed by our lab group named VenOmics for BD. That stands for VenOmics and Cell Signaling Environment for Studies and BioDiscoveries. To make a quick introduction about animal venoms, venomous lineages are widely distributed in the animal kingdom, and we can see that through the great number of animals that developed some sort of envenomation mechanism. And these substances are highly variable, being composed of many different peptides, proteins, salts, and other molecular compounds. And nowadays, animal venoms represent one of the main drivers for the discovery of novel natural products with interesting biotechnological properties, such as therapeutic, medicinal, and agricultural. Here I've put some example of drugs developed through the study of animal venoms, such as anti-snake venom produced at Butantan Institute, captopril that is used to treat high blood pressure, and a strong analgesic named Prialt that was developed through the study of a sea snail venom. With the importance that animal venoms represent, here we represent the Venomics and cell signaling environment for biodiscovery, or Venomics for BD. That is a novel web-based public database under development for omics storage and integration of multi-level venomous data, such as transcriptomics and proteomics, that can be derived from venomous and envenomated organisms. And it also works as a platform for integrative analysis to conduct the knowledge discovery process. The platform implements several experimental, computational, and statistical approaches of systems biology to discover molecular targets and mechanisms of action of different venom components. Venomics for BD comes up to improve genomics research, serving as a common place for data storage and downstream analysis of heterogeneous biological data. Here, put as an example a common workflow of genomics research that starts with genome extraction or inflation, followed by multiomic sequencing and data processing. This information can promote a higher understanding of genome expression from different regulatory bottlenecks, as well as understanding the differential expression caused by envenomation inflicted animals and across different conditions. Integrated analysis can lead to a better comprehension of the molecular biology behind genome activity and action, leading to the process of biodiscovery. In the platform overview, users are able to visualize data from public projects and to create the private ones to store data from multi-level omics such as genomics, transcriptomics, and proteomics. Private projects can be shared by the respective coordinator with other users to visualize, download, and conduct multiomics downstream analysis. And Genomics for BD follows the same intuition of other popular databases to facilitate user experience. For new users, public projects are available as examples on how to create and edit your own project. Here is a print screen of Venomics BD for homepage and a pipeline of the data needed to create your own project and perform downstream analysis. With the platform, users can perform analysis across different conditions like differential protein or gene expression and integrate different regulatory levels to the analysis to give a broader overview of the biological system at study. All tools are available through graphical interfaces to facilitate the process for users not so used with the command line. And here's an overview of the analysis menu that users can choose to perform, and a print screen of the crossing tab, where users can select which experiments to use as control and treatment. Here there are some analysis results performed within the platform where I put here as an example the necessary inputs to perform several analyses, and from that we can calculate a scatter plot comparing the gene expression and respective proteins found in the transcriptome and the proteome within the same condition, and a PCA plot of expression data obtained from two omics levels, transcriptome in the left and proteome on the right. And users can also calculate pathway activity score that provides an interactive heat map and hierarchical clustering of metabolic pathways across different conditions or treatments. And here are some examples of WGCNA analysis and output visualization tools implemented in the platform, where we can see here, for example, the model trait relationship that illustrates the most relevant gene clusters for each condition, a sample dendrogram and trait heat map, and a gene tree dendrogram. So here we presented VenOmics for BD, that in its conceptual model allows to storage, manage, retrieve, and integrate experimental studies, and it's a platform developed to reduce the gap between dry and wet lab experiments. In the future, new tools and features will be implemented in the platform to improve other Venomics downstream analysis, and we hope Venomics for BD becomes a powerful resource, especially for scientists studying venomous systems of non-model venomous organisms and their signaling effects. Thank you.